Okay, cellular respiration is the process when the body converts biochemical energy from nutrients in the food we eat into energy that's used in our bodies, which is in the form of ATP, which is adenosine triphosphate. So the first step of cellular respiration is glycolysis. I think I said that right. But it is an anaerobic process, and it's basically the pathway that converts sugar into energy, which is glucose to a pyruvate. It breaks down larger molecules into smaller ones, which is a 6-carbon molecule that'll be broken down into 2 3-carbon pyruvate molecules. The energy used to split the glucose is done by the 2 molecules of ATP, which is the adenosine triphosphate. Another electron carrier of glycolysis is the NADH, which stands for nucleotide adenine diphosphate, which is the NADH, and it is used in the 3rd stage of cellular respiration. At the end of glycolysis, well, this is the NADH here. The NAD makes the NADH, which is the nice nicotinamide adenine diphosphate. Yes. And that's the 2 molecules that'll be formed at the end of glycolysis. It'll be the 2 molecules of the NADH and 2 net molecules of ATP. Not the ATP synthase, the ATP. Like I said, it was anaerobic, which means no oxygen is required. So it's anaerobic. And there will require a aerobic function later on in the 2nd stage of the cellular respiration. And that is when the pyruvate molecules the molecules produced at the end of glycolysis, they are transported into the mitochondria. In the mitochondria, they will be transformed into 2-carbon acetyl group, and it will be picked up by the coenzyme A, which is a carrier compound, and it's called an acetyl-CoA is the compound name of it. Is the compound name of coenzyme A is acetyl-CoA. And it's going to deliver the acetyl-CoA. Acetyl-CoA is going to deliver acetyl group from the pyruvate through the citric acid cycle, which is also known as the Krebs cycle, which is here. This is the Krebs cycle here. This is going to be the glycolysis taking place up here, and the process of the pyruvate molecules. Here is a detailed sample of the acetyl-CoA transporting into the citric acid cycle, which is the Krebs cycle. During the Krebs cycle, there are 4 or 5 steps, and the first one is going to be combining the 2-carbon acetyl group, the acetyl-CoA, with a 4-carbon. I'm not sure how to say it. Oxaloacetate molecule, and to form a 6-carbon molecule of citrate. Then the citrate loses a water molecule and gains another as citrate is converted into isocitrate, which is here. So it's from the citrate, then it forms into the isocitrate. In step 3, it is the isocitrate is oxidized, and which is oxygenated, and it's producing a 5-carbon molecule with a molecule of CO2 here, with a molecule of CO2 and 2 selected electrons, which reduce NAD, the NAD plus to the NADH here. These 2 steps are they're highlighted here. I mean, well, they are here. They are decarboxylation steps here, 3 and 4, and step 5 is when a phosphate group is substituted for coenzyme A and a high energy bond is formed. So the energy is used to substrate level phosphorylation to form either GTP or ATP, and the ATP is what we would use for energy. The GTP and ATP are 2 forms of the enzymes called isoenzymes, and they are found in the tissues that use large amounts of ATP, which is the heart and skeletal muscle. Step 6 of the citric acid cycle is dehydration process that converts succinate into fumarate. Succinate, and then it dehydrates it into fumarate. Water is then added to the fumarate into the next step, and malate is produced. And the last step in the citric acid cycle regenerates the oxaloacetate by oxidizing malate. This oxidizing malate right here. So that then does this. Oops, sorry. Yeah. And that will make a molecule of the NADH. And this process, the citric acid cycle process, is continuous. It is not stopped. It is just a continuous cycle at the cellular respiration, at the cellular level. Then electron, the oxidative phosphorylation, which is the final stage of the aerobic part of cellular respiration, which is the oxygenated part, and that is categorized by 2 substages: the electron transport stage and the osmosis stage. So in the electron transport stage, high energy electrons are released from NADH, which is in the yellow here, and FADH2, and they move along the electron transport chains found in the inner membrane of the mitochondria, which would be in here, in their inner membrane. It is a series of molecules that transfer electrons by chemical reactions. They are found making up 3 complexes of the ETC. Some of the energy from these electrons are used to pump hydrogen ions, which is the H plus you see here. Here, all of these great things are the H plus, the hydrogen plus ions. And it is they go from across the inner membrane here. They go from across the inner membrane from the matrix into the inner membrane. So from here into the inner membrane of the mitochondria. This is the whole thing is mitochondria. The final thing in the ETC, in the electron transport chain, is the final protein are gained by the oxygen molecule, and it is reduced to water in the matrix, which is here again, and it is reduced into water. This is the last stage, which is the Osmosis Stage. It creates a greater concentration of ions in the inner membrane space, producing an electrochemical gradient. The gradient causes ions to flow back across into the matrix where the concentration is lower. So the concentration is higher and then it gets lower. The flow of these ions through a protein complex is the ATP synthase, the adenosine triphosphate synthase. The ATP synthase acts as a channel protein, helping the hydrogen ions. They are helping the hydrogen ions here across the membrane. So they are here, and then the ATP synthase is helping them cross into the matrix. It's the flow of hydrogen ions through ATP that gives the energy for the ATP synthesis. After passing through the electron transport chain, the low energy electrons combine with oxygen to form water.